I'm Ylli Doksani. I'm head of the DNA Replication Stress Response group in IFOM. My main research interest is to understand how cells deal with replication stress, with DNA replication stress. Now, we are all familiar with the concept of genome instability and how DNA damage can cause genome instability. We know that our DNA can be damaged, and this is often inevitable. Living our lives, food, breathing, sunlight, alcohol, cigarettes, they can all cause DNA damage, and most of them are inevitable. And we know DNA damage is bad, but what makes things really worse is when you have to replicate this damaged DNA. When you replicate damaged DNA, then you have a serious risk of fixing permanently this damage as mutations, or even worse, causing chromosomal aberrations, changing the structure of your chromosomes. Now, this has been known for a long time. It's known that DNA replication is the moment of higher vulnerability of our genomes. And this is a major field of study in trying to understand the causes and consequences of genome instability. But the problem is that it's complicated, right? So it's complicated because you have to study transitions that occur on the DNA, not just proteins, but also transitions that occur at the level of DNA structures. And this often requires site-specific studies, so to look for events that happen at a specific site of the DNA. And because of this, most of what we know about the DNA replication stress response comes from studies done in budding yeast. Budding yeast has been invaluable, and still is an invaluable tool to study DNA replication stress. And the reason is because in order to understand how something is happening, a mechanism or a pathway, you have to catch it in the act, which means in space and time, and space being exactly the place on the DNA where the problem is occurring. Now, while this is relatively easy to do in yeast, it's much more complicated to do in mammalian cells, which we ultimately want to understand. So while synchronization can help with the time, to find the right time where to study this, the mammalian genomes are way bigger and way more complicated and less known, or less characterized than yeast genome, which makes it much harder to study to look for site-specific events in the mammalian genome. Now, what I and my group want to bring new in the field is to offer a new place where to study this replication stress response in mammalian cells. So a site where this happens, and that is telomeres. Now, telomeres are famous because of their shortening, because they shorten every time our cells divide, which ultimately determines how much we can regenerate our cells, and this directly influences aging and also cancer. Now, telomeres, apart from this, are also a very good tool to study DNA replication stress response for three reasons. One, because there's many of them. We're not looking now for just one side of the huge mammalian genome, but we have 2 copies for each chromosome, so there's roughly hundreds of them per genome. The second thing is because of their sequence, they are a very good substrate for studying DNA structures. They are easy to label, and they are good for fluorescence microscopy and also for electron microscopy studies of DNA structure. And third, and most important, is that telomeres are known to be part of a category of genomic sites that are called replication fragile sites, which are sites that have the highest probability of undergoing DNA replication stress. So this makes telomeres a perfect system to study replication stress response in mammalian cells and to understand really the molecular mechanisms that are in play during replication problems. So the connections with cancer research are in understanding the pathways that operate in maintaining genome instability, or the pathways that are inefficient or that can go wrong in cancer cells. So understanding better these pathways means that we can also target them better. We can design better drugs, how to target them, and hopefully know what to look for in different patients and in setting up personalized medicine. So which pathways can change in different patients and how to target them properly. This is one aspect. The other aspect of studying, or the implication of studying replication stress response is the effect of chemotherapeutic agents. Many chemotherapeutics agents are basically DNA damaging drugs, and they ultimately interfere with DNA replication, right? So often we know that one DNA damaging drug works better than the other, but we don't know exactly why. And I think that the secret relies in what type of stress they precisely cause. So studying how they interfere with DNA replication and what mechanisms the cell operates to respond to these drugs can help us understand better how they kill cells or how resistance can occur with these drugs.